Hello, everyone. Welcome to our webinar today, where we will explore the many advantages of light sheet theta microscopy and how it can help you advance your research. My name is Peter Lang, product manager here at MBF Bioscience, and we would like to thank you for taking your time today to view our presentation. I'm honored to introduce you to our primary speaker for today's webinar, Dr. Raju Tomer, inventor of light sheet theta microscopy and assistant professor of biological sciences at Columbia University, where his focus is development and application of molecular, optical, and data analytical methods for multi-scale understanding of complex biological systems. Dr. Tomer has been working in microscopy development over a decade and has made pioneering contributions in developing several highly effective microscopy systems. Raju, you want to greet our audience?
Hey, hello everyone. Thank you for joining.
Just a few words regarding the agenda and some technical details for today. Professor Tomer will provide some general background on light sheet fluorescence microscopy and clearing techniques, followed by a comprehensive overview of the unique advantages of light sheet theta microscopy, as well as example images using this powerful technique. We will then discuss the next generation implementation of Dr. Tomer's innovative light sheet theta microscope, namely MBF Biosciences' ClearScope, and show an overview of the system operation. Feel free to submit any questions using the Q&A option at the bottom of your screen. We have set aside time after the presentation to answer those questions. If we're unable to get to your questions, we'll be sure to get in touch with you directly to discuss in more detail after the webinar. All right, so let's get started. I'm going to switch things over to Professor Tomer.
Great, thank you so much, Peter, and thank you everyone for joining today. Good morning, according to time in New York here. So let me start my screen. Okay. Right, my screen visible?
Yes, it is.
Okay, great. All right, so thank you so much for joining again. So in this presentation, I'll generally discuss the light sheet microscopy and its use for imaging large clear samples with high resolution, and with a specific focus on the advantages light sheet theta microscopy, this new implementation of light sheet microscopy concept that provides over the conventional light sheet system for imaging much larger samples. So let me start by just highlighting that over the last decade or so, there has been a lot of progress made in the so-called tissue clearing field. So as you would know, or many of you would know, that biological tissue generally tend to be very opaque. So you can only image them to limited depth, right? So with these advances in these various methods, which use cocktails of certain chemicals and processes, these opaque biological tissues can be turned transparent, right? So like mouse brain or embryos or any other organs, right? So there's been a lot of work done over the last decade, and many different methods have popped up with their pros and cons. So different methods providing different advantages. Some are good for immunolabeling purposes, and some are very good in providing much better tissue clearing and hence optical access. So anyhow, at the end of it, you end up with this highly transparent tissue, which is optically accessible, right? So here's an example of a cleared mouse brain processed with a particular method called clarity. So on the right, you can see it's completely transparent. You can completely see through it, and it's generally optically homogeneous, but still inhomogeneity at the micro level, though, as we will see. So this new capability has also presented a lot of challenges when it comes to imaging these samples. Right? So if you want to image such large sample at high resolution with conventional methods, let's say confocal point scanning, that's going to take a very long amount of time, weeks and months, and especially if you want to do high resolution imaging. So in that context, about a decade ago, we sort of optimized or adopted this light sheet microscopy idea for high resolution imaging of such large, clear, transparent samples. So light sheet microscopy, just very briefly, it uses a thin sheet of light. My mouse is visible, right, Peter?
Yes.
So the basic idea is that we create a thin sheet of light which illuminates the sample or the plane that we are trying to image, right? And then it's only that plane that is emitted signal, which is collected by a standard wide field detection arm, right? So this provides us optical sectioning, so we can image the entire plane with a fast CMOS camera, as opposed to point scanning in confocal or two-photon microscopy. Right? So we end up with a couple of orders of magnitude faster imaging speed. So we spent quite some time in optimizing implementation of light sheet microscopy for high resolution imaging of large clear sample. And it presented its own challenges when we are imaging larger samples. So going much deeper than few hundred microns, so the optical properties changes and so on. So we had to make quite some effort in optimizing the optical path or from sample to all the way to camera, and also work out other strategies like adaptive parameter correction, so that with that we can sort of correct for any deviations between in the light sheet and the focal plane alignment that may result because of the large depths that we are trying to image. So that allowed us to do sort of high resolution imaging of mouse brains or samples of similar size that are cleared. So here are a couple of examples from old work. Depending on the biological question you have, you can choose the label. So on the left side, it's an example of using viral tracers to sort of do anatomic tracing of connections from prefrontal cortex all the way to hippocampus. On the right side, you see an example of using the so-called immediate early genes, which are the genes that become activated in response to neuronal activity. So mapping those genes at whole brain level, you can get a snapshot of the sort of let's say the functional state of the brain. So by now, it has become fairly regular to image that kind of samples with in a matter of hours. Right? Just to also demonstrate sort of from resolution perspective, so light sheet microscopy based approaches can get you high resolution at the large scale, right? So we can develop these sort of Google map like views of the brain, that we have this complete brain, and then we can zoom in down to individual dendritic spines level of details. So for last few years, we have been asking the question that can we scale up these imaging approaches to samples larger than mouse brains? So current standard light sheet microscopy is pretty much limited to samples that are sort of roughly similar to mouse brain size, which is about 1 centimeter wide, 6-7 millimeter in thickness, right? So we have been working on trying to scale up these imaging capabilities to much larger samples. For example, right, like a centimeter thick slab of human brain, right? So if we have methods to make them transparent and label them, then we need to be able to image them to take full advantage of that, right? So there are a lot of work happening on the tissue clearing and labeling front. Already, we can reasonably clear up to centimeter thick pieces of these tissues, so like human brain or other larger brain samples. So methods. There's still a lot of challenges on the labeling front to label them with antibodies or other reagents, but it's getting there, right? So from the imaging side, it also presents another set of challenges in the light sheet microscopy context. Right? So if you look at the light sheet microscopy, so it has an orthogonal geometry. You have this light sheets that you are generating 90 degree to the detection arm, and they need to travel through the tissue to illuminate the plane that you are trying to image, right? So that puts hard limitation, first of all, on the sample size. So of course, the sample cannot be bigger than the working distance that these objectives allow. But even then, even if you have smaller than the working distance, you have optical limitation because light sheet is traveling through this tissue. And even though it's clear and so on, there's still a lot of scattering happening. And by the end, you sort of reaches towards the center, you lose much of the quality. So here's an example to demonstrate that point. So it's about 5 millimeter thick section of rat brains, approximately 2 centimeter wide. And you can see it's as clear as you can get it. So it's optically accessible as best as we can make it. And so here we used a label for vasculature, which is kind of present all over the brain. And if you image this sample by light sheet coming from side, so from left side, let's say in this example, you end up with this kind of scenario. You have higher signal, higher quality on the edges, which are near, but as the light sheet needs to penetrate deeper in towards the center, you lose the quality, even to the extent in the center, in this case, you don't see anything, right? So even without the physical limitation of the working distance, there are optical limitation on how deep we can illuminate with a sheet of light without suffering in the quality and the signal. So we have been trying to solve that problem. So one easy way or nice way is to sort of rotate the geometry 45 degree, so that you're sort of 45 degree relative to the sample surface. So here's let's say a slab of large sample we're trying to image. So if we sort of put our illumination objective, detection objective 45 degree, then we sort of solve this problem of having to come through the side of the samples, right? So that in general works very well, especially for low NA detection objective, which generally have longer working distance. But the couple of issues, I think the significant issues are that one is that you are sort of chewing up in your available working distance, right? So especially if you want to use high NA objective for high resolution imaging, you can't afford to lose a significant part of your working distance to be able to image deeper, right? Before you run into the sample itself. And then the second issue, which I think is significant also, is that you are sort of imaging an oblique plane. So a plane, different part of which are sort of exposed to different amount of tissue, right? So if we are trying to image this plane, different part, the signal coming from different part will have to travel different amount of tissue, right? So that introduces different aberration, different errors in the same plane itself, which is very hard to correct for in general. So that also becomes significant, especially if the sample is not ideally clear, which is most often the case, right? It's not completely clear, it is, let's say, partially clear, then it will lead to reduction in quality. So we have been exploring an alternative idea while still maintaining detection quality. So we want to be able to detect perpendicular to the sample surface, right? So we developed this idea that if we generate the thin sheet of light, and instead of coming at 90 degree as a standard configuration, if we come at an angle which is less than 90 degree, and hence that's why we called it light sheet theta microscopy, just to sort of highlight that it's less than 90 degree angle. Then we can reach our detection plane with this thin sheet of lights, which intersect the detection plane in a line illumination, right? And then with this optics trick, we can quickly scan that plane, scan that line in the detection plane, and hence achieve planar imaging. So it preserves the speed of light sheet microscopy. So most light sheet systems use the scan beam, scan light sheet, which are essentially a line beam which is going up and down fast. So it provides similar imaging speed. We're still doing detection perpendicular to the sample surface. We are removing the limitations on the sample dimensions, at least in the lateral dimensions. And of course, there's still possible limitations from the working distance of the detection objective itself, that we cannot go deeper than that, right? So this conceptually provides or solve some of these problems that we encounter when we want to scale up these imaging approaches while maintaining the high quality and high speed. There are, of course, several other implementation trying to look at that problem using different kind of approaches, right? So in our hand, this has worked out very well. So I want to illustrate that in the next few slides. Just first, a little bit technically how we are doing the scanning. So the idea here is that you have this blue light, which are we using to generate this thin sheet, right? And it intersects with this focal plane. That's the focal plane here. Intersect the focal plane in a line which is illuminated, which produces signal. And then we sort of scan this line in the focal plane in synchrony with the rolling shutter in the camera chip. So this SCMOS chip, we can take image by sort of in a rolling shutter mode, where you only expose few rows at a time, which you can roll. And so if we do that in synchrony with the illumination, we can remove any out of focus light that may come otherwise, right, if you don't do this synchronization. So we could use, of course, one-sided illumination, but we can also have light sheet coming from two sides, which provides further advantages. One being, if let's say sample less than ideal, if it has some obstruction on the surface from one side, the light sheet is not able to penetrate, then the other light sheet can still reach there and do the job of illumination, right? And the second thing is that we are kind of effectively increasing the effective NA of illumination. So we can end up with thinner sheets or thinner or much better, let's say, axial resolution. So so that's the way to scan it. So there are two modes that we can scan this light sheet. So one simplest thing to do is that we just use a galvo scanner to sort of move the light sheet perpendicular to its propagation direction. And in that way, we can illuminate different part of the plane. The problem, of course, there is then the different part of the light sheet will intersect with the plane. And as we know, as some of us will know, light sheet has this trade-off between light sheet thickness and the field of view, or the extent to which it is uniform, right? So that will then lead to differential quality on the edges of the image, right? So other scanning approach that we innovated in this system is the simultaneous two-axis scan. So here the idea is that we move the light sheet perpendicular to itself, but also along its propagation direction. And if we do that in a synchronous way, certain way, then we can make sure that only the same thinnest part of the light sheet is illuminating the entire plane. So we can get this high quality throughout the plane, as opposed to losing quality on the edges, which is also the case in standard light sheet implementation. But this, more importantly, this also allows us to now use higher NA for light sheet illumination to generate thinner sheets, because we are not limited by that trade-off. So we can get much higher axial resolution as well. Okay, so I'll demonstrate some examples sort of supporting these points. Before that, here's the model that we had implemented for first demonstration in our lab. Just to briefly highlight that this is a standard detection. You have an objective tube lens camera. This is a standard wide field detection arm. And then this is the arm generating thin sheet of light. And I just want to point out a couple of elements to the ETL. So we use these electrically tunable lenses, which allow us to move the light sheet along its propagation direction, so we can change its focus. And then we have this galvo scanner, which allows us to move light sheet perpendicular to its propagation direction. So if we use them in synchrony, we can do this 2-axis scan of using the thinnest part of the sheet to illuminate the plane. Another thing I want to highlight is that we have the flexibility of changing that angle. And in general, we want to maximize this angle. So the best quality, of course, best axial resolution you will get when you are 90 degrees, like in a standard light sheet. So here we want to maximize that angle as much as we can without running into the sample itself to sort of end up chewing up the available working distance, which of course limits the depth that we can image in that sample. So this is also different than, let's say, other systems which use single objective for also generating this oblique plane. They have this hard limitation of that angle that you can use, right? So here, since we have illumination coming from an independent objective, we have an independent control on the light sheet properties and this angle that we can use to illuminate and hence sort of get to a sweet spot of best quality while being able to image very large samples. All right, so some data points to demonstrate these capabilities. So here's imaging of a piece of human brain, which is stained with DAPI, labeling all the nucleus. This showing one volume. So the left is image with 1-axis scan, where we are only sort of moving light sheet perpendicular to its propagation direction using galvo scanner. And the right side is with this simultaneous 2-axis scan approach that allows to use the same thinnest part of the sheet throughout the plane. And the Z direction is the vertical direction, so we are imaging the sample from the top. So as you would expect in the 1-axis scan, similar to a standard light sheet system, you end up with differential quality. So you have this center with very high quality, where you have the thinnest part of the sheet, but as you move away from the center, then the light sheet is getting thicker, right? So you have a much poorer axial resolution. Whereas in the 2-axis, simultaneous 2-axis scan, we can make sure we get uniform quality throughout the volume, one volume that we are trying to image. So we get sort of a very good optical sectioning or axial resolution throughout the volume. Okay, so now if you revisit the example that I used to introduce the problem, this 2 centimeter wide, 5 millimeter thick piece of rat brain clear, right? So if we image with the, as we said, standard light sheet microscopy, you run into the problem of signal getting reduced towards the center. But now if we image the same sample with the light sheet Theta microscopy, we can image the entire sample with uniform high quality all across, right? And there is, of course, no limitation on the sample size. Sample could be as large as possible, as long as we have sample stages that can travel that distance. So this panel B is just showing one tile, one example tile, showing the axial resolution, axial detail. So it's image from the top, and you see here is the images at different depth, showing the high quality that we get throughout the depth there. All right, so with this system, then we can image regular samples as well, like cleared mouse brain with attached spinal cord as standard applications. So here's showing some zoom in. So this is the side view. So this is the Z-axis showing the axial resolution or the optical sectioning capabilities, and you can see the sort of a uniform quality throughout, right, in the center and in the periphery. And it's a Taiwan EYFP brain. The brain sample is from Taiwan EYFP transgenic animals. In those animals, essentially EYFP is expressed in majority of neurons in the brain, but not all of them. Here's another example demonstrating the high quality throughout the sample. So that's a thick, I think 5 millimeter thick section of mouse brain. Again, the Taiwan EYFP mice, and here you see the zoom in at different positions, the peripheral position, the central position, and the sort of more denser regions, showing the uniform quality that light sheet theta microscopy approach provides at the speed of light sheet microscopy, while removing the limitations of sample sizes on the lateral dimensions. So here's it's my favorite example. So that's imaging of a expanded section of mouse brain, again Taiwan EYFP transgenic line. Some of you might be familiar. There's a lot of effort being made in the direction of sort of physically expanding the tissue, which essentially leads to enhancement of resolution because you are sort of magnifying the tissue itself. So it can provide much higher resolution even with the standard NA detection, but of course at the cost of now even larger samples. So it's a section of mouse brain which is 4 times expanded, and you end up with a sample which is like 3.32 centimeter wide and 1 centimeter in this dimension, right? So it's really large sample, and it will be very hard to image with, let's say, confocal. Probably will take months to even just scan through it. So with the sort of this unique capability of light sheet theta microscopy, we can image that entire very large sample at high resolution with the speed of light sheet microscopy, which is a couple of orders of magnitude faster than, let's say, the confocal systems. So as you zoom in, you can start seeing all sorts of very tiny details. So these are neurons. You're currently seeing the cortex on the up of the video, and you can see various projections. So one downside of this kind of capability is that you end up producing tons of data. So this supposedly small part of the brain, but 4 times expanded, it's resulted in approximately 6.4 terabyte data. And this is something we image with only 10x magnification, 10x 0.6 NA, right? So if you use even higher magnification, higher NA, you will have a humongous amount of data. So that's 6.4 terabyte. It's even hard to visualize. So this rendering was done with 8 by 8 fold downsample data just to be able to visualize it. But if you look at the raw quality that we get out from light sheet theta microscopy with a 10x detection objective, you can very nicely see very tiny details like this dendritic spines all across throughout the sample with high quality. Okay, so beyond clear tissue, this kind of geometry also provide advantage for other kinds of sample, especially in the live imaging context. So this is one of my favorite examples, worked in collaboration with Rafa Eustace's lab. So they have a project with Hydra, which is kind of a jellyfish, right? Where they have transgenic animals that express GCAM, which is a label for neuronal activity. So we could do this live calcium high-speed imaging and sort of correlate the neuronal activity with sort of freely behaving animal. So these kind of sample present challenges for standard light sheet configuration because it's changing shape. And if you are using sort of orthogonal illumination detection, depending on the shape, the compression of the animal, your light sheet may bend different amounts. So you really need to make a lot of effort on adaptive correction or hardware-based correction in a standard light sheet geometry. Whereas with the light sheet theta microscopy, since we are coming only from one axis, other 2 axes are free for us, for the animal to move or expand into, because that doesn't affect any of the optical properties of the system. Right? So here this movie is showing the top half, the bottom half, so you can do this fast volumetric calcium imaging as an example of non-cleared tissue imaging. All right, so that was the first generation of light sheet theta microscopy that we developed. And then over the last few years, we have been collaborating with MBF Biosciences to generate this second generation system. And we have specifically focused on two aspects. So we wanted to sort of make this system compatible with multiple types of tissue clearing methods. So some of you may know there are several different tissue clearing methods with different advantages, and they also have different optical properties. So they might have different refractive indexes, right? So for example, clarity-based tissue are like around 1.45 in refractive index, whereas iDisco-based are like 1.52, and so there are significant differences. So if you have a system which is optimized for clarity from hardware perspective, right? And if you want to use it to image an iDisco sample, that's going to be a problem because there's a huge refractive index. And then we have focused, so we have come up with strategy to make the system independent of type of clearing methods used, so that we can image any type of cleared tissue with high quality. And then we've also focused on enhancing the resolution to get to a really high resolution. But that, I think, is an area where light sheet theta microscopy, which is now implemented as ClearScope, really shine in providing this high resolution for very large samples. So yeah, so that's the sort of goals of the second generation. So optimal compatibility with multiple clearing methods. And then, of course, ease of use, robust implementation, and much higher resolution. So that's the implementation from MBF Biosciences that they call ClearScope. It's really nice, fantastic, robust. We sort of held them in the context of this NIH SBIR grant, and as part of that, we also got fortunately a first prototype of the system that we have been playing with and having fun with, and now we're kind of regularly using it for various kinds of applications. I just want to give you a sense from a few application perspective, but before that, let me just say a thing about this multi-immersion capability of this ClearScope or next generation LSTM. So as I mentioned, that if your system is optimized for clarity samples and you want to image an iDisco sample now, you run into this kind of problem. As you go deeper into the sample, because of the refractive index mismatch, you just simply lose focus very quickly, right? So you can, of course, adjust the system hardware to be compatible with iDisco, but then it won't remain easily compatible with the clarity-based system, right? So that sort of requires adjustment using appropriate objectives, alignment, and stuff. So we sort of tested many different things, and it turned out by using this linearly adaptive scanning strategy, we can fix that. So this is schematic showing how we are acquiring volume, and it turned out if we just find the optimal parameter on the top of the sample and at the bottom of the sample, generally with a linear interpolation of those values work fine in giving us good quality. So it's kind of minimal adaptation required. Just generally 2 points, determining best parameter, the 2 points top and the bottom is sufficient to get us good quality throughout. So now just revisiting the same example, now the right side is the same sample imaged with this LAS, linearly adaptive scanning, applied to the imaging. Okay, let's play it one more time. Oh, it refuses to play. Okay, all right, so, but you get the idea. All right, so in the next few slides, I want to show a couple of applications that we have been using ClearScope prototype for in collaboration with other labs with specific biological questions. So first example I want to highlight is a collaboration with Jaita Basu's lab at NYU. So they are very interested in sort of understanding various disease mechanisms, various mechanisms of brain diseases, especially focusing on epilepsy and Alzheimer's. So as part of their experimentation, typically they're getting these chunks of human brain from neurosurgeons, right, from diseased patients, and they are doing electrophysiological recording from a few of these neurons, trying to figure out what is wrong in the system, right? And at the end of that, they are able to fill those neurons with certain dye, right? So to label them, which then can be visualized. So, so you end up with these large chunks of tissues, which are still large, right? And which has a few of these neurons labeled. So the next goal there really is to be able to image them in very high level of detail so that we can reconstruct the morphology and sort of reconcile the electrophysiological recording with physical models of these neurons and try to learn what's going wrong in various diseases. So here's one example showing such data set. So we of course there's no limitation on the lateral dimension of the sample that we can image, and we can acquire this. I don't know what video is a little bit jerking there, but anyhow, so we can image these very large samples with that kind of details that we can reconstruct the entire morphological properties. And eventually, of course, the goal is to develop these three-dimensional models. So that's segmented with this NeuroLucida 360. Yeah. So thanks, Peter. Peter did that. Peter and colleagues. So yeah. So then we can reconstruct these neurons in human brain, right? That we have recorded from, or our collaborators have recorded from, and then try to correlate that deficits with the morphological deficit, and of course in the context of overall circuitry that, of course, we can fill in more detail. So with the ClearScope or light sheet theta microscopy system, we can do that, right? So if you use, let's say, confocal microscopy, it will take you ages to acquire that kind of data. With light sheet theta microscopy being a couple of orders of magnitude faster, we can these samples become accessible in a reasonable time frame. Another example is to demonstrate this multi-color imaging capabilities. So this is work from PhD student in the lab, Yanan, is developing ways to label and map vasculature in human brains. So he's just showing one example. It's a large chunk of human brain, as you can see from the scale bar, which is 1 millimeter. It's centimeter wide, and of course there's no limit on the sample size, right? And he has stained them with 2 different antibodies which are labeling 2 different types of vasculature, larger and small ones, and so we can perform this multicolor imaging with ClearScope and the light sheet theta microscope. All right, so final example I want to show from a non-neuroscience field. So that's our collaboration with Lars Dietrich, who's a faculty in our department. PhD student Hannah is working on these biofilms of bacteria and trying to understand various aspects of them, also related to their pathogenicity and so on. And Shraddha from my lab helped with the imaging, or helped with the sort of establishing the imaging assay, right? Because whenever you have a new sample, it takes some time to figure out the mounting strategy and so on, various parameters, right? But with the capability that ClearScope or light sheet theta microscopy provides, right? There's no limitation on the sample dimension. Lateral dimensions, these biofilms can grow to several centimeters, right? And they are bacteria, so you really need high resolution to see details of various problems that occur in these biofilms. So these kind of samples are also accessible, right? So beyond neuroscience application. And of course, various different organs that can be cleared, or biopsies from clinical world can be imaged at high resolution and high speed. All right, so with that, we'll just sum up. So try to convey that this variant of light sheet microscopy, light sheet Theta microscopy, which is implemented as ClearScope, the next generation of it, it allows us to do high resolution imaging of very large samples. So essentially no practical limit on the lateral dimensions of the sample, only limited by the travel range of the stages. It's compatible with multiple types of tissue clearing method without having to do any hardware change. So at the software level, we can do this linearly adaptive scanning, which can image samples from clarity to all the way to iDisco with different refractive indexes, making sure we get the best quality throughout. Right. So eventually, so that sort of paints the picture that we can image that kind of large sample, like a centimeter thick slab of human brain with that kind of resolution. That's in terms of capability, but of course, to be able to do that, it also presents other challenges, especially on the data side. Right? It probably is going to be petabyte or even beyond scale with that kind of resolution. Right? So to be able to do that. All right, okay, so with that, I will stop. I want to acknowledge all the people in my lab. So light sheet Theta microscopy work, which I described, was done by Malika and a former PhD student, Bianca. I also mentioned work by Yanan on vasculature mapping. Shraddha is our staff engineer. She is really the person running the ClearScope and sort of exploring new applications, along with Chang. Chang is also working on that system. And also in the context of today's presentation, I want to also acknowledge Dan, our lab manager, who has helped with a lot of sample preparation and so on. And thank you to my MBF colleagues. It has been really great interacting in the context of SBIR grant, with a lot of back and forth and visits and so on. And we are very happy that we got a chance to get this first prototype and really play with it. And I want to thank our collaborators, as I mentioned in the talk, and our funding support. And I want to thank you so much for taking the time and listening to the webinar today. Thank you.
That is great. Thank you, Raj. Just to kind of review some of the topics that Professor Tomer hit on, we'll switch gears here and look at MBF Biosciences implementation of Professor Tomer's patented LSTM technology. Yeah, ClearScope is basically your complete light sheet imaging solution. For those users who want to experiment with imaging parameters or explore innovative ways of imaging clear tissue specimens, we have an advanced user mode that provides precise control over all the hardware components. But to make it accessible, we also have a basic user mode for simplified operation, which includes automated light sheet optimization tools and streamlined workflows directly in the software. We'll take a look at some of those tools during sample acquisition video in a moment. But really, ClearScope was designed with automation and efficiency in mind to help researchers increase scientific throughput without having to be an expert in optics or hardware control. A highly versatile 3D visualization environment, advanced stitching capability, and a full suite of optimization tools with state-of-the-art functionalities are all included to support your analysis and publication needs. ClearScope essentially enables you to obtain quantifiable imaging data pertaining to neurons, vessels, other subcellular structures like dendritic spines. The imaging flexibility, as demonstrated by the numerous samples using various clearing protocols in Professor Tomer's talk, enables you to choose the best clearing method for your specific application. We've coined this intelligent refractive index compensation, and it's performed automatically by adjusting the depth of the light sheet as a function of the refractive index change and tissue depth. So it effectively enables single detector objective to image specimens cleared with various clearing protocols, like Clarity, Cubic, iDisco, et cetera. users can simply select the configuration directly within the GUI. And ClearScope itself is compatible with many different types of detection objectives, from leading lens manufacturers, ranging right now. We offer objectives from 4x to 25x with various numerical apertures, anywhere from 0.28 to 1.0. And we'll continue to add new options as lenses come to market. Yeah, but really the idea is that the modular hardware design of the system allows for scanning of large biologic structures at a variety of magnifications. And we can actually do so at up to 7 laser wavelengths, which are completely customizable to your research needs. NBF Bioscience is really working with research labs in academia, as well as industry, to develop cutting-edge image processing solutions that dramatically speed up multi-terabyte image stitching, as Professor Tomer mentioned. That's kind of the big issue. You collect all this data, and now you have to process it, view it, and analyze it. ClearScope can be coupled with NBF's leading data analysis programs, such as NeuroLucida 360 for neuron reconstructions, that we investigated clear tissue for quantifying your tissue, VesaLucida 360 for quantifying vasculature. And essentially, all these programs enable you to collect better image data, produce higher impact publications, and advance your research. So let's take a closer look at the operation of ClearScope itself. So essentially, this is our user interface here. And I'll just review some of the tools that we have here. So we have a very intuitive ribbon bar tool set up here, which basically groups tools of similar functionality into one location, so it's very easy for you to navigate. So for instance, in this case, we're looking at the Acquire tab. So we have options to look at live image, launch tissue scanning workflow, take stats, etc. On the right-hand side, we have a series of 2 panels with camera controls, setting up your multi-channel acquisition, what channels you're interested in actually acquiring, laser controls to adjust intensity during the acquisition, as well as light sheet settings. So we make all these settings and tools available to you, as I said, as part of that advanced imaging mode. However, as discussed previously, ClearScope software includes these automated light sheet optimization tools and streamlined workflows to really help researchers increase their scientific throughput without having to be experts in optics or hardware control. So we essentially click the auto alignment button under light sheet settings toolbar to start the light sheet optimization process. System will perform a number of steps, such as determining the galvanometer, scanning start and stop points for the synchronization with the rolling shutter exposure of the camera, and automatically adjust the ETL settings, ensuring only the thinnest part of the light sheet intersects the detection plane. The software will repeat this process for each arm and each channel. So we're typically using multicolor bead slide for alignment and calibration purposes. This is helpful to assess the imaging performance of the ClearScope system. So we're going to set up a quick multi-channel acquisition using the stack acquisition tool here at the bottom. In this case, we've configured a 2-channel 30 micron acquisition with half micron Z-step. And the Z-meter here is going to indicate our position as we perform the scan. Once completed, we can evaluate the stack using the quad view mode of the 3D visualization window. Given our red and green channel images are well registered, the resulting max projection view of these beads should appear spherical in profile and yellow in color, as we see here. All right, now let's load a sample. The sample we're going to be looking at today was provided by Professor Tomer. It is human brain tissue that was immunostained and cleared with iDisco. Let's see. So we're going to go to the load position first. So we have controls here to easily go between the load position and the focus position. And then we're going to start the slide scanning, the tissue scanning workflow rather. Let's see. This provides step-by-step guidance for acquisition of your clear tissue sample. In the first step, we choose the scan type. There are 3 modes: scan extents of place markers, which we're going to choose here, scan a defined volume, or scan basically a grid scan. And you have the option to perform a pre-scan over the defined region of interest, where the system will utilize an automatic tissue detection routine to mark only tile locations where tissue is present. This is basically to optimize the acquisition time as well as resources needed for storage. After completing setup, we'll click the next step. At this point, we can select a channel from the multi-channel control tool and begin to place markers to define the region of interest of our scan. We are prompted to place markers to define the left, top, right, and bottom edges of the region of interest. And the macro view over here shows us an overview of our scan. And you could use this to easily navigate to locations in the region of interest to perform the next step in our workflow, which is basically to set top and bottom Z positions. So here, what we're going to do is go to a location and then focus above the sample, click the set button in the workflow, and then we're going to focus through the sample to the bottom and click set to define the bottom extent of our scan. Now, as Professor Tomer mentioned, we can apply this linear adaptive correction, we call it intelligent refractive index compensation, to automatically adjust the depth of the light sheet as a function of the change in the refractive index. And that is done. Here we'll adjust the galvo position and the ETL for optimal image quality for each of the 2 arms at 2 positions, near the top and bottom, and ClearScope will automatically adjust the light sheet parameters, interpolating values throughout the Z range. So this is just done automatically here for you as we scan through the Z position. So we'll see here, we'll check the box to apply the interpolation map, and then we can scan through, and those parameters are automatically adjusted. Now to the final step here, we're going to define the location and save the image and the file type. By default, we use a JPX format. It's uses efficient data compression to minimize data storage requirements and maximize speed. TIFF output is also available. Near the bottom of the workflow here, you will see a scan summary providing pertinent information such as the required disk space, number of channels, size of the area. As you start the scanning, a dialog window shows the scan progress. And upon completion of the scan, a preview of the data is instantly available. You could jump through the focus positions to inspect the image data and choose the option to compile individual stacks into a single file, if you like. Image data may then be used in our built-in 3D visualization environment, or opened in one of our quantitative analysis software tools to produce accurate data for analyzing vasculature, morphometry, cell populations, or other subcellular processes. If you have any tissue samples you'd like us to image with ClearScope, please feel free to contact us. We'll be happy to arrange an imaging section so you can take a look at how your tissue can be imaged using this revolutionary technology. So that was just a quick overview of our ClearScope operation. I know Professor Tomer went into a lot more detail with respect to the science behind things. At this point, what I'd like to do is open things up to your questions that have come in. Let's see. So, our first question: what is the maximum thickness of a tissue you have scanned using this technique? both in mouse and humans? Also, can we image monkey brain with this? Well, I can tell you, MBF Biosciences side of imaging, we've done 6 millimeter thickness in mouse brain, and we've done some human samples, a few millimeters. Raju, maybe you can.
Yeah, well, so I mean, the working distance, of course, let's say that this Nikon objective that you have, it provides 8 millimeter, right?
Correct, yes.
So I mean, we have image up to 6-7 millimeter of like a piece of monkey brain that was cleared.
Okay, great.
So in general, I would think if the clearing has worked out well, then you can go as deep as your working distance allows, right? So we have image 6, 7, 8 millimeter deep. So it can maintain the quality, especially with this adaptive parameter correction strategy, right? Which could take care of the loss in the properties.
Right, great, thank you. Let's see, we have a second question here. Have you used clearing protocols preserving endogenous fluorescence reporter signals, GFP, RFP, M-Cherry, or Rainbow?
Okay, I can answer that. Well, so different clearing methods have different strengths. So, for example, iDisco method, right, which is good from the perspective of using antibodies to label, but it also ends up killing the endogenous signal from GFP and RFP and so on. But then there are other methods like Clarity, which preserves very well these fluorophores, so endogenous fluorophore. So I presented several examples of the brains, right? Taiwan EYFP, Taiwan GFP that we image on the regular basis. So yeah, depending on that boils down to choosing the right clearing protocol. If you're interested in more using antibodies to label specific molecules, then it's let's say iDisco, at least in our hand, gives good results compared to other methods.
All right, great, thank you. Here's another question, and I believe this is for you, Professor Tomer. What clearing method do you currently use, realizing there are different methods and selection is somewhat dependent on starting tissue? So what is your preferred method and why, I guess?
Yeah, I mean, in our lab, we regularly use at least 2 different methods, Clarity and iDisco, simply because these two are sort of complementary in a way. One is very good for antibody labeling, other is very good for preserving the fluorophore and stuff, right? So we generally use Clarity and iDisco, but there are a lot of different methods around, and every few months there's a new method coming up, right, with different properties. So I think it makes sense to spend some time in really figuring out which method is best for you. But the advantage with the light sheet ClearScope is that it's kind of independent of clearing method they use. So it could, without any hardware, without any need for hardware change. So that gives us the flexibility.
Let's see, maybe we got time for one more question. We are running close to our time limit here. Let's see, for multi-channel imaging, what are the time costs associated with adding additional channels? Since there is a single detector, I'm assuming each channel must be collected sequentially.
Yeah, I guess currently that's the case, right, Peter?
Yeah, yeah. I mean, basically, for multi-channel imaging, we do need to collect them sequentially, but we do utilize a typically multipass filters, so that the switching is really a matter of changing the excitation wavelength. So we try to maximize the switching speed by rapidly switching the excitation wave lasers. Let's see. All right. As I said, I think we are coming up close to the end of our time limit here. What we can, I do want to. Can
I comment on this live mouse?
Oh, sure, absolutely, absolutely.
That's a So that's, I mean, in our custom light sheet theta system, we had the capability of live imaging, which is essentially easy to add, right? We just repeat the experiment again and again. I mean, in terms of advantage of microscopy for live imaging, let's say live mouse embryos, I feel it's a very good system to do that, especially mouse embryos are not transparent as like zebrafish larvae or even Drosophila embryos, which are still accessible. So mouse embryos present its own challenges, and if you can manage not to come from side, but with the sort of light sheet theta, then it can provide advantages. But as I understand, I think that's not yet implemented in ClearScope, right? The live imaging capability.
That is correct. That is not implemented as of yet, but it is on our development roadmap.
Okay.
All right. Let's see. First off, I would like to thank Professor Tomer for joining us today. I really appreciate you taking the time out of your day to do this with us, and you've been a valuable resource for the development of this project. I'd like to thank the team here at NBF Bioscience and the NIMH for their funding support. I'd like to thank you for joining us, taking time out of your schedules today. I hope you found the presentation informative. If you are interested in learning more about ClearScope or any of our other products, please contact us at info@nbfbioscience.com. Please don't forget to fill out the short survey at the conclusion of this webinar. Your feedback is a very important component to our continuous improvement process. For general discussions, FAQs, general imaging, anything from acquisition to analysis to worm tracking or multi-photon imaging, we do have forums where you can communicate with each other and NBF experts at forums.nbfbioscience.com. This presentation has been recorded and will be available for future viewing on our website, nbfbioscience.com webinars. And thank you all again. We hope to see you back again in the future. Don't hesitate to contact us in the future. And we look forward to hearing from you. All right. Take care and thank you very much again.
Yeah, thank you. Thank you everyone for joining.